Thank you for the introduction. Hi everyone, my name is Felipe Melis. I'm from Chile, South America. Um, and the title of my presentation it will be Gene Regulatory Network for Infant Bifidobacterium species. So I'm going to shift a little bit the topic to bacteria. I want to start to ask you a question for yourself. How many of you are aware how important is the environment shaping our microbiome? It's well known that diet can directly affect the gut microbiota, modulating its composition or metabolic output, promoting health or also diseases. And also this impact in the way that the microbiome interacts with their host. So if we add medicine, host diseases, or even pet interaction, we have a complex mix of factors that give shape to our microbiome. So among these key factors that I mentioned before, we have the diet, also pharmaceuticals, geography, and even life cycle stages. I want to focus on this, a stage that all we are here already past, that is when we are babies or infants. It's the key factor that modulates that stage could be among others delivery mode, that could be by vaginal birth or C-section, and also the type of feeding, that could be mother's milk or formula. As a fact, for my country, 50% of the deliveries in Chile are by C-section, rising to a 70% in the private system. If we go to the WHO, the World Health Organization, they say that this must be between 10 to 15%. So one of these motive me to try to contribute to the knowledge of this. So what are the members of this so-called infant microbiome? Well, I focus in Bifidobacterium genus. They belong to the Actinobacteria phyla. They are gram-positive and anaerobic. They also are known as a health promoter bacteria, and their abundance and biodiversity is shaped, as I mentioned before, by delivery type or type of feeding. There are uses as probiotics, and they have a very good saccharolytic capacity. These species of this genus have different adaptations to their ecological niche. These adaptations, among others, could be the use of EPS, that are exopolysaccharides, that gives them advantage to adverse condition, and as well promoting human health. They also have pili, promoting adhesion to the intestinal wall, and also they do the degradation of HMOs. HMO stands for human milk oligosaccharide. That is a complex mix of sugars that are part of the mother's milk, and they almost exclusively consumed by species of this genus. So, as I mentioned, breastfeeding is a key factor for shaping the infant microbiome. So, in the work here of Larsen et al., they analyzed the microbiome of different infants that were breastfed, and their microbiome is completely dominated by one species, that is Bifidobacterium longum subspecies infantis, or B. infantis. And I also wanted to notice in this point, where the infant was fed with formula, and their microbiome was completely reshaped to other species of Bifidobacterium, only for just consuming formula. So it's important how the environment is like shaping the microbiome of their babies. So to contribute to the knowledge of this, I wanted to harness the power of databases. Use the most available data that we have from these public databases. I collect genomes for 4 species of Bifidobacterium. In this case, Bifidobacterium longus subspecies infantis, Bifidobacterium longus subspecies longum, Bifidobacterium brevi, and Bifidobacterium adolescentis. Through passing a quality analysis of those genomes for my downstream analysis, in order to build pangenome, a pangenome is a shared collection of the genes that are part of a group or a species or a strain, and also the genes that are shared by a specific group inside of the strain. And also I use those genomes, to build gene regulatory networks in order to know how are the relationship between the transcription factors and the genes that are regulated. So I wanted to start with this. This is a metric of, it's a quality metric of a huge number of genomes that I use. So in the blue line is the genome length, and the red line is the M50. M50 is often a quality metric used often to assess the quality of a genome, of a genome assembly. So in this point, when I pointing with the arrow, we have a M50 that is equal to genome length. That's good because maybe it's a reference genome or it's a genome in one context. But also I wanted to notice that these points here, we have lower genome lengths and also lower M50. So continuing with this, I want to give the message that always have like quality analysis in our pipelines, because it's not always using all the genomes available. So always we have to look at the data and see if there is like problem in them, because in some cases we have very poorly sequenced genomes. So continuing, after I did my quality analysis of our genomes, I get a high quality number of genomes for these 4 species, and I build a pangenome, that is this collection of shared genes among the species for each one of those species that I use, and I collect the upstream region of those orthologs, the promoter region, in order to use it for scanning for transcription factor binding sites. For each one of those, I build a consensus upstream region for those orthologs that are found using the pangenome approach. I scan for transcription factor binding site, and I build the relationship between the transcription factor and the target gene. So I use the information from clock classification, the ortholog prevalence also, if an ortholog is present in all the species or a subgroup of those species, and also the score of the motif that gives me the scanning. So for building the pangenome, I use an approach of all against all ortholog comparison. So I generate n number of groups of orthologs for each one of the genomes, of each one of the species, and I get a core genome that is in terms all the genes that are shared by the same strain, and also an accessory genome that are genes that are shared only for a few members of that group of species. I extract the consensus upstream region in order to scan for transcription factor binding sites. So first, I wanted to know if those pangenomes, this shared collection of genes, are open or closed. So based on this relationship between the number of genes and the number of genomes, if we look at the slope of this plot, we can know if these are closed or open. So based on this structure of this plot show that these pangenomes are open. What this means, if we sequence new genomes, probably we will find new novel genes for these species that we are analyzing. But also that this means that we have still a space to continue exploring these organisms. So if we continue sequencing, probably we'll find new genes. So also I want to talk about this pangenome size metric. This is the number or proportion of genes that are part of the pangenome, or all the genes that are present in these strains. So when I was reviewing this slide, I noticed that, for example, B. longum, subspecies longum, or B. longum, has the bigger pangenome, also has the biggest proportion of cloud genes, that are the genes that are shared only by 15% of the species or less. So when I was reviewing this, I think that maybe when I collect this genome, I collect some genomes that are not related to that are not related to be longum subspecies longum, so maybe I have some sort of strain contamination in the genomes that I collect. So the thing what I did was use those ortholog groups and extract the promoter region, and I scan for transcription factor binding site, use position scoring specific matrix in order to build the relationships that I have between the transcription factor and the target gene. So I wanted to show this first network from this forest strain. Here I have this network that is divided by accessory genomes and core genome. And the core genome, here are the species, are the genes that are shared by 95% or more of the species. And the accessory genome, I want to focus on these three transcription factors that are related to the consumption of HMOs by these bacteria. So one of the things I saw that I have many connections of FUCR, that's a transcription factor related to the consumption of fucosylated HMOs, and these many connections to the accessory genome, maybe they are telling me that there is some sort of differentiation in the pathways that are using these bacteria to metabolize those sugars. So it's, according to the literature, we have difference in the strains at the level of consuming these HMO. So maybe these connections, also I have more connections for the accessory genome than the core genome. And also I saw the same for NACR, that is a transcription factor related to consumption of other type of HMO, and I didn't see this for NANR. Continuing with the Bifidobacterium breve, I see the same for FUCR, this transcription factor, but I saw many connections for both, for accessory and core genome. So it's well known in literature also that breve has the capacity of degrade this, but is related or associated to a strain specific so maybe this is telling me that also. I saw that for NANR I have the same pattern, that but for NACR I didn't see the same many connections. For Bifidobacterium longum subspecies longum, they only have the transcription of NACR, but I also have like many connections to the accessory genome and a few connections for the core genome. Also for adolescentes is different. They lack the capabilities of the other three to consume HMOs, so I use this as a contrast for the other three. For the other three, yes. Finally, I want to give the message that I'm trying always using the most available data, always have a strong Q&A processes, and also I want to test this in genome scale models that are mathematical representation of the reaction inside of a cell. So I wanted to know, I have to go to see more details, what are the connections that I have for those transcription factors. Also, I wanted to say thanks to my advisor, Daniel Garrido, in Chile, to Arita Sensei, that was so kind to receive me in his lab in Japan, and also to ANIT, that is the government foundation that funds me, my research and my travel. Thank you.
Thank you for the very interesting talk. We now invite for questions.
Hi, thanks for your great talk. So I have a question, it's more for technique. So I saw you show a lot of a great gene regulatory network plus. So I'm wondering which program in the package you're using for draw it?
All of this, for this is Python. Yes, Python.
So you use a package when you do the network? And
yes,
then they
for the network, basically, the thing that I did was using a package from Python for the scanning for transcription factor, for scanning motif.
Okay.
That scan a motif inside of genome, and then when I have the relationship, I use Python to build the network using network X. I think this is the right name of the package. Yes.
Okay, thank you.
Thank you for the nice talk. I have just small questions. So, as far as I know, HMO is quite variant from mother to mother, right? So, in case of your bacteria species analysis, did you consider about the HMO variation between the mother to mother and then onto the geographical region and so on?
Sorry, can you repeat the question?
HMO.
Yes, sorry.
Yeah, this one is quite variant from mother to mother.
Yes.
So, did you consider that kind of factor when you analyze about this large scale of the data? Because in case of the bacteria species communication, community, and so on, you need to consider about the individuality.
Yes.
In case of the HMO, as far as I know, this is quite variant from mother to mother. So I just want to know what kind of factor you consider.
Maybe that relationship from mother to mother is we can see in the different strain that I collect from the public databases, because you have different connections for different species. So that species was sequenced by a particular baby, maybe, because of that is that they have this difference in contrast with the others. So maybe that difference, the HMO from mother to mother, is, I can see that in the specific strain that is different from the other one.
Okay, thank you.
Okay.
Hello, thank you for the talk. Just a very curious question. You show very well how much there is a difference of gut microbiome between the breastfeeding baby and the not breastfeeding baby. I was just wondering how much this difference is actually maintained in the adulthood, or it's impacted by the adult diary?
Well, actually, this predominance of Bifidobacterium is only from babies to infant. It's like when we are babies or infant, we have a dominance of like 60% of Bifidobacterium in our organism, but after that, Bifidobacterium decreases to 1% or less. So one of the challenges that exists right now is trying to use this as a probiotic in adults, but the difficulty is that if we eat the probiotic of Bifidobacterium, it's difficult to maintain it in the body because we are not consuming mother's milk. So that's the challenge right now, trying to be able to thrive that bacteria in our gut, but as an adult. So there is many challenges. In infants, they are using probiotics with infantis, and they are really good. So the challenges are with us as adults.
Thank you.
Okay, you're welcome.
Just one quick question for my own curiosity. So I'm just wondering whether you see that sort of difference in skin microbiome as well, on top of gut microbiome?
Yes, you can see those different. Yes, the environment is shaping all the microbiomes. So skin microbiome certainly it's also impacted by
different
the
method?
yes.